Continue. Okay, so Emma, Adi, and Yashis are here. And no Edward yet. And then Kathy is online. And Max is online. She told me he was going to. All right, well, anyway. So by the end of today, hopefully you'll be able to model more complicated selective scenarios. Yesterday, we got to dive really deep into how to model pretty complicated demographic scenarios, but not necessarily, we were kind of sitting on the surface with respect to how to model selection. So we're going to get into more complicated selective scenarios. We're also going to talk about how to optimize your simulations and run them from the command line, because while GUIs are really fun to play with and they have cool colors, they are not the most efficient way to run Slim. So today we're going to talk about modeling complex selection, and then we'll talk about scaling up. After I talk about modeling complex selection, we're going to take a break, and that's when the quiz is going to happen. And I'll also show you, so I've been giving you little exit tickets every day. The workshop also has a, like my official course evaluation that they will use to decide if they want to bring me back in the future. So please say I did a good job, but also give me like stuff to work on for the future, because I read all of your comments and take them into consideration. But anyway, I'll do that in the middle. And then in the second half of the day, we'll talk about scaling Slim up, and some of the ways you can do that and some of the problems with doing that. So with that, a selective sweep is when an adaptive mutation arises in a population and then sweeps or increases in frequency until it is very common. Yesterday, especially, I was talking a lot about fixation, which is when when an adaptive variant reaches a frequency of exactly 100% of the population. We can also call fixation a complete sweep. You might instead decide to have your simulations end at a partial sweep. In nature, oftentimes sweeps end in at a partial frequency. They're not complete because of a variety of different complicated real world demographic situations, and also just the fact that selection is always ongoing. So you might want to model what a partial sweep looks like. In these cases, you're going to need to change your ending condition. So rather than ending your simulation at a set time point, we're going to end the simulation dynamically depending on what the frequency of your adaptive variant is in the population. So in this particular block of code, I'm saying from generation 10,000 to 100,000, every single generation within this late block, I want to check the frequency of the adaptive variant, which is the M3 freak. So here I'm taking, looking at the mutation sim.mutation frequencies of all mutations of type 3 and taking the sum of those. Why am I doing it that way? Well, if you are allowing this mutation 3 or whatever your adaptive locus is called to evolve multiple times through random de novo mutation, you're going to have actually a bunch of different M3 mutations because they originated at different times and in different individuals. And so if you just take the max, or if you take the average, in fact, you're not going to capture the true frequency of the M3 allele over the course of the whole population. You're going to be taking the average of all of these variants, or all of these different kind of independent evolutions of this variant. If you are, so the more independent evolutions of a mutation you have, the more important this is. And then I just take the sum of that. So sum sim.mutations frequencies, sim.mutations of type M3, and then an if statement. If that frequency is greater than 50%, I'm going to end my simulation there, and maybe I would also include a sim.output full to include the output there. So all of this is to say that you can change this if statement to be any frequency you want. If you want to do it 90% or 100%. Now, if you wanted to do a complete sweep and you had not turned off convert substitutions, this bit of code would not work because if you'll recall, when a mutation type reaches a frequency of 100% in the population, SLIM automatically will convert it to a substitution and just get rid of it, which means that this particular approach won't work if you're looking at a complete sweep. And instead, I didn't actually type it out, but in the manual, there's language around this. You can instead say, is the frequency of the M3 mutation equal to exactly 0? And if that's the case, that means that it's either was lost or it's recently been converted to a substitution. And then you can check whether how many M3s have been converted to substitutions, because it does keep track of that elsewhere. So that's a different way of doing it in order to look at full sweeps. So one way we can classify selective sweeps is based on what our ending condition is. The other way we can classify selective sweeps is based on how they start. So we can also classify sweeps based on whether they are hard or soft. This has nothing to do with their ending frequencies, but rather how they're, like I said, how they're started. Hard sweeps tend to be very slow. In a hard sweep, these are adapted from a class I taught on the evolution of infectious diseases, so it is very much related to antibiotic resistance evolution. So let's say that you have some kind of virus and you start taking antibiotics, and it makes the treatment very efficient, and so it drops the population size super small. And now it has to wait, you have to wait until a brand new mutation shows up. And there's a long waiting time until you get that beneficial mutation. But you only get one individual in the population, one individual viral particle gets that adaptive mutation, and then it sweeps that single drug resistant strain, starts to increase in frequency. So a hard sweep is one or very few origins. They tend to be very slow because you have to wait a long period of time for a mutation, at least in terms of this kind of framing around antibiotic treatments or antiretroviral treatments. And also, if you look at the genotypes after the sweep, you'll notice that because we started here and the sweep happened relatively quickly, basically everyone in the entire population has the same genotype. There was only a single origin, and so nearby mutations that were linked that didn't have a chance to get broken up with via recombination or whatever hitchhiked along with it. And so in the entire population post-sweep, you see a reduction in diversity, because not only at this selective locus, but also around the locus, you're going to have only one or two, maybe very few genotypes present. Soft sweeps tend to be faster. In a soft sweep, we're referring to when there are many or different origins, or just multiple different origins for the adaptive mutation. So let's say that there is a less effective treatment, which leads to a wider bottleneck. And so 3 different strains of viruses all happen on the same mutation at around the same time. And they're all in parallel rising in frequency. Now, at the individual adaptive locus, they're all the same, but around that adaptive locus, they're all different. This is also one of the reasons why burn-ins are important, is to generate that diversity around an adaptive locus. And so post-sweep, it might look like this, where strain 1 has this genotype, strain 3 has this genotype, strain 2 has this one, and strain 3 has this one. And so the diversity around the locus, whoops. There we go. The diversity around the locus actually has a much smaller decrease post-sweep, or it stays the same. And so in a soft sweep, beneficial mutations rise in frequency, but it's not that one individual strain is reaching fixation. Now, overall, we can still say that actually in this particular case, the adaptive locus reached fixation, but no single strain is becoming dominant. We can define hard and soft sweeps using a term we call theta. Theta stands for the population scaled mutation rate. I feel like I'm missing a slide here. Okay, so I don't know what happened here. Okay, so theta is equal to 4 times the population size times the mutation rate. So what is theta telling us? Just looking at kind of like what we're multiplying here. Yeah.
It's sort of a measure of like how many mutations in the population.
Yeah, exactly. So it's about how many mutations are going to occur per generation. So if theta is greater than 1, that means more than 1 mutation will happen per generation. And so we're probably going to get a soft sweep. Whereas if theta is a lot less than 1, that means you're going to get very few. The probability of getting the adaptive mutation in a single generation is relatively low. And so they are probably going to get very few examples. Yeah.
You said like if it's a lot less than 1, but like it's not a lot less than.
Yeah. So the breakpoint is considered to be 1, but there's a lot of work by population geneticists that show that if it's 0.99, that's a basic, like, this is kind of back to this essentially neutral, where that's kind of a fuzzy border. So you can have a theta that's quite, like, again, a theta of 0.99, it's basically a theta of 1, which is basically a soft sweep. In general, when people model soft sweep, or when people model hard sweeps, they're using a theta of 0.01. That's kind of like a general rule of thumb. Because, like I said, if let's say you have a theta of 0.1, that means that you're going to get an adaptive mutation every 10 generations, give or take, which is going to happen that is going to happen more frequently than, say, the time it takes for a single adaptive mutation to rise in frequency. Now, it's probable that if one mutation has started to rise in frequency and another one shows up, like that first one is at higher frequency and it'll probably crowd the other one out. But it kind of gets fuzzy. Like these hard and soft lines it's a useful dichotomy, but it's not a perfect one. It's also because with soft sweeps and desire, or just generally desire. Yeah, so that's totally true. So what I will say is that the probability that a sweep gets broken up has to do with recombination rate. So if you make recombination rate dependent on mutation rate, absolutely. What I will say is that so there's a bunch of different things that can control whether or not a haplotype gets broken up, or whether or not the beneficial mutation gets broken up with its hitchhikers. So one of them is how long the sweep takes. So if you have a really super duper adaptive mutation that is going to rise in frequency super duper fast once it shows up, then there's not enough time for recombination or mutation to happen. Whereas if you have a really weakly adapted mutation, it might take longer, and that's more time for recombination to break apart. There's like some work around like hard sweeps turning soft, where like if they get to intermediate frequencies, sometimes they'll break themselves up through recombination, and then they turn soft. Yeah, but for sure. One way to make sure it's actually hard is to use add drawn mutation in SLIM. So there's 2 different ways that you can kind of introduce an adaptive locus. One is by using the sim tags that I was talking about, where we're rate and then turning it on after a certain point. The other is after the burn-in, you add a new drawn mutation and you see if it evolves or if it doesn't. We'll talk about that a little bit more. But so there are many ways we can get a lower theta, or actually there's 2, because there's 2 parameters here. So one of them is population size. So hard sweeps tend to correspond to small populations and or lower mutation rates. And there tends to be a long waiting time after an environmental change until the novel adaptive genetic diversity appears. Soft sweeps, on the other hand, have a higher theta, which means either you have a large population size, lots of individuals to happen upon a mutation, or a high mutation rate, a high probability that any one individual has that new mutation. And also it tends to be that soft sweeps come from populations that have higher standing genetic diversity before a sweep. So sometimes, especially in infectious disease research, when you're talking about selective sweeps, it might be that a mutation was neutral before, but then you turn on a selective event, and all of a sudden that mutation that happened to be just randomly neutral in the population becomes adaptive. And so soft sweeps can often originate from those kinds of scenarios because multiple individuals, or that neutral locus that becomes beneficial might be on multiple backgrounds. So yeah, theta is the predicted number of mutations at a locus per generation. And as theta approaches 0, or as theta gets smaller and smaller, it becomes more unlikely that a second mutation can show up. I like to show this graph to think about hard and soft sweeps. So if we're looking at the first half of the pandemic, do you think, and we're looking at the strains, you can kind of see selective sweeps happening where we started at one strain, the 614G strain, and then over time, the 614G strain became the most common one in the population. These are, okay, it's kind of strain with, it's both. I would say that the reason why you see strain replacement is that there were like adaptations. This is like a whole thing, but in general, infectious diseases evolve very rapidly and tend to evolve to be less deadly and more infective over time. So if you want to, as a virus or as a disease, you want to be not super deadly, because if you're deadly, you're going to kill people too fast and then they can't infect other people. So there's this kind of like, there's a really common trend where over time diseases evolve to be slightly less deadly, but they try to like minimize deadliness while maximizing infectivity. So there's a lot of like different selective pressures that COVID-19 especially was under. Some of those included trying to be less deadly but more effective. Others included later on in the pandemic, vaccine escape mutations, or not necessarily just vaccine escape mutations, but adaptive immunity escape mutations. So as people got one strain, then they became pseudo protected against it. But yeah, I think of strain replacement as when, as a colonization ecological scenario in terms of the gut microbiome. So strain replacement is the idea that you have some strain and then a brand new strain comes from an outside source and colonizes it. These are evolving from each other, and we can make a very coherent phylogenetic tree. I don't know off the top of my head what the differences are. I know that Omicron is like way different than everything else, but I can't tell you off the top of my head. But the point I'm trying to make here is in the first half of the pandemic, before say January 2021, does that look like a hard sweep or a soft sweep? Yeah, so this here.
That looks like a soft.
Why does it look like a soft? Why does this look like a soft sweep to you?
Oh, wait, where? Oh, sorry, I was looking where it's at like January 2021.
All right, let's look at like, I guess, October 21 back.
Oh, that's
in the first like part of the pandemic, first few months of the pandemic.
That looks like a hard sweep.
Okay, yeah. What is the, yeah, a little bit before January. What does the second part of this graph look like? Soft sweep, right? So there's multiple different strains all originating, especially right here. There's like several strains all originating and some rising to intermediate frequencies.
So with strain, even though they aren't like absolutely the same way, what I'm saying is like
No, okay. So in the previous graph, when I was showing you a cartoon of hard and soft selective sweeps, they were all evolving the same locus. That's not quite true here. This is kind of an illustrative cartoon because I want to point out that, okay, this at least looks like a, whoops. Earlier on in the pandemic, it looked more like hard sweeps. Why was that, do you think, versus later in the pandemic? Like, what was changing about the ecology of COVID-19 later in the pandemic that maybe led to more frequent soft sweeps? Okay, vaccines. Why would vaccines lead to soft sweeps? Okay, I would say that like vaccines and increased selective pressures are probably not like a change in selective pressure doesn't have to do with hard versus soft. Looking at this equation, what's going on?
It could be the case though that different loci aren't going to also be another user.
Sure.
Well, I think the population size is drastically increased.
Yeah. So before,
early 2021.
yeah, so in early 2021, the number of individuals who had COVID-19 was a lot significantly smaller. I think January 2021, or maybe it's like April or July. I think it's like July of 2021 is like the largest peak. When I was making this graph for an undergrad class, you're all asking more advanced class questions than they did. The point I was trying to make here was that there's a lot higher population size, and that could be one of the contributors to why we see more strains evolving independently. Another answer could be that there's been more time for COVID-19 to diversify and spread across the entire globe. And so there's higher standing genetic diversity. So those are kind of two of the reasons I was thinking of. But some of the things you threw out were also correct. I don't think vaccines, please prove me wrong. Send me a paper later. But I don't think like differences in selective pressures necessarily, well, so given the like previous things I was telling you about, if you had a vaccine, a really acute selective pressure is actually more likely to generate a hard sweep because you're more likely to kill everything off and lead to a huge bottleneck in population sizes. So hard sweeps are also kind of about like founder effects, if that makes sense, where like you're only having a couple of founders. So in the sudden contraction of population size by a really effective vaccine, that would maybe be the case. Yeah. Sure. Yeah, there are multiple. Yeah. Yeah. So
that's just America. There's
yeah.
AstraZeneca.
Uh-huh.
Other parts of the world. So those were
I don't remember enough about the early COVID vaccines. I believe they were all using the same sequence, or they were all using the same strain. It was about like the stuff around the mRNA, and I'm not a vaccine person, so, or I mean, I did just get my COVID shot on Sunday, but like I'm not I'm not an expert on vaccine science, but I do like them. Is what I was trying to say. Anyway, yeah, there's I would be curious to read more about that, but in general.
Also, those areas.
Sure, yeah. So there's more time, like part of standing genetic variation getting built up includes the fact that recombination is breaking apart adaptive loci and putting them on new backgrounds. So that, yeah, that could also be I want to say like the COVID-19 phylogenetic tree looks like this, where like everything is over here and then Omicron is over here. At least that was the phylogeny like a year and a half ago. Like Omicron is really different.
How much genetic selection is there
How much what?
Is there, do you know, like, the difference between positive selection and negative selection?
Yeah, that's a really good question. I don't know so you're kind of asking like what the distribution of fitness effects in viruses kind of is. So like so for example, in humans, for everybody else in the class, in humans, deleterious mutations are the most common and beneficial mutations are pretty rare. I think across the tree of life, that is pretty true because it's very easy to fuck up function, but it's a lot harder to innovate. But I don't know. That's a good question. Google it for me.
Yeah. I was thinking about it as like for negative selection on vaccines.
Yeah, sure. I will also say in general that most antibiotic resistance mutations are deleterious in the absence of antibiotics. And so I would assume that the same is true for vaccine escape mutations, or something similar might be true, where there's this like trade-off that you're investing in. Although vaccine escape is well, vaccine escape is the same as immune escape, which, yeah, anyway. Yeah, yeah.
So we're trying to trace the history around that. Like the area is actually much higher
Yeah. 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 Maybe I'll can do like an infectious disease version of this class. So to connect things back to SLIM, which is supposed to be what this class is about, you can use just this simple theta expression to model hard versus soft sweeps. So this is also a place where you can practice, or where I was practicing defining constants. You can use, if you want to define a constant, you simply do that in initialize. And then throughout the rest of the code, I can use theta and that will equal 1. So if I make theta equal to 1, that means I have a soft sweep. And then I'm saying that I want the mutation rate at my beneficial locus to be equal to theta over 4 times n. But in this particular case, I'm making the mutation rate for all other mutations equal to 1e to the negative, or to 10 to the negative 6. In this particular case, I don't want there to be a ton of deleterious and neutral mutations, because the more mutations that there are, the slower my code is going to be. I only want to toggle on the hard versus soft for the beneficial locus. And I'll say M1 are neutral mutations, M2 are deleterious, M3 are beneficial. And then I initialize 2 different genomic element types. So in the G1 type, I'm allowing neutral and deleterious mutations, which deleterious mutations are 2.3 times more likely to occur. This is from the human distribution of fitness effects, I think. Actually, no, this is from the microbes. It's, yeah. I think that's, yeah. The DFE it has nothing to do with that. No, you're right. Thank you. And then I'm making a genomic element type called M3. And essentially, I'm just making a single locus where the adaptive variant can go. And the rest, and so there's only one place for that adaptive variant. I'm ensuring that there's only one version of it, or that there's only one place for it to go. There's a single locus. But by toggling or by jacking up the mutation rate, I can still allow there to be multiple different origins. They're just going to all be the exact same adaptation or place in the genome. And so then I make the genomic element G1 0 to 500. I make G2 a single locus somewhere in the middle, and then the rest goes back to G1. And then I can have the mutation rate vary by providing it a vector of different mutation rates and the different break points I want it to go. So this is saying that up until position 500, I want mutation rate to be mu. At 502, I want it to be mu ben. And then for the rest, I want it to be mu again. So you can see here throughout that I'm using symbolic constants that I've defined later on in the code. And you can also, this is also a demonstration of how you can create a mutation rate that varies. So grab a slim piece of code and we're going to practice modeling hard and soft sweeps. So you can, again, you can take what you wrote. I think everybody wrote an adaptive variant version yesterday. So take that and put the mutation rate for the beneficial locus equal to theta, or scaled to be equivalent based on theta. And run your code keep all of your other parameters the same. Run it once with a theta of something very small like 0.01 or 0.001, and once with a much higher theta of like 1 to 3. Remember to include a burn-in, because if you don't include a burn-in, you're not going to have any sort of diversity pre-sweep. It's just going to like appear as soon as you start to do it. And I want you to calculate heterozygosity at the end of the burn-in, right before you start allowing selection to occur. And then I want you to calculate heterozygosity after the mutation has reached a frequency of 50% or more. And so use calc heterozygosity, which we used a lot yesterday, to look at diversity. And our goal is to recapitulate these two figures. Okay, so we're trying to calculate heterozygosity pre-sweep and post-sweep, or after the burn-in and after the beneficial mutation has risen to high frequency. You can choose to have it be a complete sweep or a partial sweep. I'm going to do the same thing I did yesterday and make a table on the whiteboard. And once you finish your things, you can add that in there. But again, the burn-in is important because if you don't do a burn-in at all, pre-sweep diversity will be 0. So we need to generate some amount of pre-sweep diversity. Any questions? Yeah.
Do you recommend using the drawn mutation here? Or?
So I would not recommend using add-drawn mutation here, because add-drawn mutation is great for hard sweeps. Okay. But well, to be fair, you can make it, all right, let me go back a second. If you'll recall, the probability that a neutral mutation is lost in the first generation is around 35%. The probability of beneficial mutation that is like has a reasonable selection, like a realistic selection coefficient, is lost in the first generation is also around 30%. So when you do add new drawn mutation, there's a very, very good chance that it will just go extinct very quickly. In fact, I think like a probability of fixation or a probability of establishment that is like indicative of a beneficial mutation just needs to be greater than, for a population size of 10 to the 4th, just needs to be greater than 10 to the negative 4th. Okay. So like we're talking about very small probabilities things actually fix. There's a bunch of tricks you can do, but anyway, the point is that if you want to reuse the same code to do both a hard and a soft sweep, putting a recurrent mutation and putting it in terms of theta is useful. The second reason that is useful is if your add new drawn mutation goes extinct in 10 to 100 generations, a new one will show up. Okay, I'm going to make the table and then start walking around and answering questions if you have it. Yes, I will go back and show you the previous slide. You can also open, like, I wrote this. Go to the Google Slides and copy and paste it. Make it whatever you want. I don't care about any of your parameters other than theta. Sure. I mean, you can use, so this is also, what I will say is that if you copy and paste this is incomplete because there is no recombination rate. Oftentimes when I'm doing these coding blocks here in class, I wrote only what I needed to and not what you would actually, or what I'm trying to highlight, and not necessarily what you need to run it. So good debugging practice, I guess. Yeah. I would say I think you'll get the same results whether or not you do deleterious or not. But have fun. You know, the only thing I care about is that you have a single adaptive locus and that it evolves with a rate that is proportional to theta, and that you run this once as a hard sweep with a theta less than 1, and once as a soft sweep with a theta greater than 1. You don't necessarily. You can also plot post-sweep haplotypes, although the thing I would like to get is heterozygosity before, at the end of the burn-in and then after the sweep. And like I said, feel free to copy and paste the code and adapt the code I have on the previous slide and like 2 slides ago where I was talking about ending a sweep. Mm-hmm. Yeah. So the sim tag is one way of doing, yeah. Definitely need to have these, but they can be either controlled. The termination statement code is
Oh, the termination.
Yeah.
Oh, do you mean like the?
It's just so cool, so fun.
Well, there's recipe.
But it should, I don't know, it should be. Yeah, so I would recommend Frankensteining together the code that was on the last slide. A couple of slides ago when I was talking about ending partial versus complete sweeps. And then the code from yesterday about syntax. I think if you just like Frankenstein that stuff together, you should basically get the right thing.
We can also sort of do it right now. So what's, for anyone who's gotten to this point, what is this initialized statement missing that you would actually need to include? Okay, yeah. So initialize recombination. Sure. One negative, I'm gonna say 7. We're also missing n, so we need to define constant up here. Oh, Max is already finished. Cool.
In the initialize, you also have to add the.
No, so syntag goes after this. So the syntag does not go inside of the initialized comment. Initialize goes, you would do, is its kind of own thing. It's hard to read, but that's the wrong syntax. Oh, perfect. Thank you, Max. Okay. So in this case, it would be something like this. We'll go afterwards. Then I would go 1 early. She can't even see this anymore. So after initialize, you might say something like mutation. You would need to use the syntax, and then you'd want to go 1 early sim dot add sub pop. Is it just sub pop or subpopulation? And then sim tag. Zero. Yeah. It makes the quotations weird. I'm sorry. Yeah.
No, it's not you.
Yeah. Somebody else was having, Kathy on Zoom was having. You do not need to name the subpopulation anything. Oh. No, you need to call it P1, huh? Yeah, you do need to call it something. Just kidding. So print calc heterozygote, or I would probably do print. Huh? Don't even, so one of the things that you're going to find out is that it doesn't actually keep true the joy of stochastic simulations is that you're not going to get the same thing every time, and that's okay. What do you think? Yeah, that's kind of what you should get, but if you don't, that's okay. Huh?
Oh, okay. In the code from yesterday, we no, no, the code from the few slides up.
Gotcha. Yeah. So that's at the end. So I'm just creating the end of the burn-in.
Oh, okay.
So end of burn-in. Run the haplosomes. Well, I probably want pre-sweep. So this little bit of code right here is saying let's end the burn-in and output this, and then we might want to we'll add something like this.
So when you say 100,000 codes or whatever gets there, they don't start with this one.
So
the simulation
I see.
would
Well,
finish
okay.
before it even gets to 100,000.
That's true. Well, okay, so I haven't updated these numbers yet.
Oh, okay. So
But-
if you do like 100,000.
Yeah. So I would say, okay, so at the moment, this is saying that my burn-in ends at generation 10,000. And so I would want to not start trying to end my simulation until the generation after that. And maybe give it a few extra generations. So this is I think how I would probably code it up, right? So this is the initialize that we just went through. And then here on this next slide, I'm defining a sim tag to make sure I can turn on and off my sweeping mutation. Here I am saying at the start of my burn-in, I want a sim tag equal to 0, so no beneficial mutations. And then here, this is saying generation 10,000 early is the end of my burn-in. And so I want to print out my heterozygosity pre-sweep and turn on the sim tag. And then for every generation after that, I need to check to see what the frequency of the adaptive variant is. And if the adaptive variant is high frequency, I will end the simulation and print out my heterozygosity. I'm going to copy over.
Also,
Yeah.
I think this is Slim 4.
Yeah.
I think Slim 5 doesn't have the cat in the print. You can just do the plus sign without the.
Good to know. It still does work if you say cat though.
Oh, it does. Okay.
Yeah, it doesn't freak out, but okay. Yeah, so Max, one thing is that you gave me one heterozygosity score. Was that pre or post sweep? And can you give me, like I said, I want to see both before and after. So Emma's got huh? Yes. Gotcha. Pre and post sweep. So again, what we're trying to do is generate this figure here as a class. Where before, so after the burn-in or before the sweep starts, what's your diversity? And then after the sweep is over, what's your diversity? There are markers like behind the whiteboard right there, but there's more right back here. You do not need to give me a ton of significant figures. As an FYI. You're all being very, very, like, very important. You're going like all the way out to a lot of decimal points, and I don't actually need that. This is kind of a. Uh-huh. Yeah, so I mean that's what this shows. So your diversity should stay about the same. You might see a small reduction because there's still like there's gonna be some dominant haplotypes coming out. I used 1,000. You can use whatever you want. I don't think it is it's not super important to me at this juncture what simulation parameters you're using outside of that you run this once with theta and of 0.01 or 0.001 and once with a theta of 3. Yeah. So what are stochastic simulations? Sure, partial sweeps. Yeah, so maybe try switching it from a partial sweep to a complete sweep and see what happens. Oh, so if you had beneficial mutations from the beginning, that's definitely going to give you weird stuff. So a couple of things that I want to highlight here is I also did this exercise, and like some of the times I did get an increase in diversity, but most of the time I got a decrease. Yeah. So partially it's because of partial sweeps. Partially it's because it's stochastic. My husband hates when I say just it's stochastic. But like random chance means that your simulations are going to give you slightly different results every time. And even if all of you in the entire room used the exact same parameters, we would get a spread of different results. Okay, let's see. So Max says pre 0.005 and then 0.006. Yeah, and you do not need to cherry pick your results by any means. Please, post one of the ones where it didn't work out. But one of the reasons why we're going to move towards running SLIM in the command line is that you need to be able to run your simulations hundreds or thousands of times and then bring together your results across many simulations in order to find the trends you're looking for. Okay, and this was 0.0. Okay, cool, Max. I got yours. Well, Max is to show the pattern what you would see.
You want to increase it to like 0.9 for the frequency.
Yeah. There's a lot of stochasticity in simulations, and that's, I think, part of the joy of them. So when I did it, I actually did get at least one run of each that looked something like this, where the soft sweep stayed about the same and the hard sweep showed a decrease. And the haplotype plots are kind of hard to see, so don't even bother looking at them.
Yeah, great. That looks perfect. Yeah. Now try running it, changing, making the data more. So these are that looks great. Yeah.
Okay, but hopefully from this exercise, you got two things while Edward is finishing up. So first, hopefully you understand the difference between hard and soft sweeps and how we can use theta to tell us that information, and also how we can hopefully some information about how you can normalize theta across different population sizes to give you the same expectation for the number of new mutations or the kind of sweep you're expecting to get. So that's kind of the first part. You should also have understood what some of the expectations are and ways you could debug your code. So you know that in general, hard sweeps should lead to a reduction in diversity. And if you're trying to model a hard sweep and you don't see a reduction in diversity, something weird might, at least half of the time, something weird might be happening. Although we all also got an appreciation right now for stochasticity and how there's going to be variance in results. That means it's not perfect. And finally, you've created a very nice simulation code that can model a selective sweep. So I'm now going to change gears and talk about context-dependent selection. So before we were talking about a single mutation that always had the same effect. Sim tags are changing whether or not we allow mutations to occur there, whereas in the future, we might want to change a mutation so that it's beneficial in some contexts, but deleterious in others. And the most common example of this is known as epistasis. So epistasis is a function of the fact that evolution sees proteins, not individual mutations. This is also the reason why we see why some mutations are synonymous and some are non-synonymous. It has to do with you have a codon and that converts to a protein coding or an amino acid change. But then the amino acids all get strung together to create a protein that is structurally very complex. And so in this like very famous like paper that first came up with was kind of the start of the idea of epistasis from the 70s, Maynard Smith mentioned that evolution is kind of like this old word game from the late 1800s, where you have some word and you're trying to change one letter at a time to go from one word to a different word. So if I want to get from word to gene, I might start by changing the D to an E, and that gives me a word that says war. Then I might change the G to, or the W to a G to get gore. And then I might go from gore to gone, and finally to gene. Now, at this particular step, I could have also gone to W-E-R-E, or where. That's still, or were is still a word, but G-E-R-E isn't a word. And so kind of central to this idea of changing protein evolution is a word where we're changing letter by letter. Also, not all words are actually really words. And that means that not all steps are equal. So the requirement of meaning corresponds to the requirement that each unit step in evolution should be from one functional protein to another. In other words, going from were to GARE, actually, I think GARE is a word, but like it's not, pretend it's not. Going from word to GERE, G-E-R-E, that's not an efficient step, or that's not a possible step because you're losing function. And the same is true for proteins. So imagine I want to go from list to poke. I want everyone to come up with on the piece of paper or just in your head, what pathway you would go with from list to poke, changing one letter at a time. In the same way that we went from word to gene. And once you have a pathway, raise your hand so you can tell me. Yeah, Brendan. Uh-huh. Yeah. Who else came up with that pathway? I think that's yeah, so I think that's list to lost. Technically, you could also go to pist, and I think pist is actually pist is a real word, and I googled this. Or you could go to, actually no, pist isn't a real word, but lyes is a real word from chemistry. But even whether or not you went from lost to lose, the next most in either way of this, you could go to lose. They both are the same kind of next step. You could also go to post or like. And then so list, lost, lose, pose, poke, I think is the most common pathway along this. But technically you could try to go to loc instead. So, yeah, so like I said, these are, if you were to list out all possible ways of getting there, these are all of the possible ways of getting there. But not all of these paths are created equally, because many of these, I guess LISC is not a real word. I guess pist is a real word. When I Googled this when I made this slide like over a year ago. And loc is also a real word. I think it's like part of a wagon or something. Maybe I'm mixing it up with yoke, but it's these are all real words, but some of them are more common than others. And so if you were to use, Google has this really fun feature where you can look at what the most common words are over time. So if I look at lies, lost, and pissed, lost is obviously the most common word. And so that's the one that all of our brains went to first as the first step on this. So most people go to lost first. And then after that, lose is probably the most common, but post I've seen a couple of people get to as well. And no one's going to LISC again. So fitness landscapes are ways we can explain mutations in a similar way, or especially multiple mutations in a similar way, where our goal is always to increase fitness. So imagine I have two mutations, one at this A locus and one at this B locus. And in this graph, this little a, little b is the wild type allele. And so I can, oh, that's wrong. So I can either have the big A, little b, these are mixed up, or the big A, big B genotypes, or, so there are 4 possible genotypes based on whether or not I have big A or little a and little b or big B. And so let's say the fitness landscape looks something like this. This is saying that if I have this little a, little b genotype, I have a fitness of 1/2. If I have this is wrong, big A, little b genotype, I have a fitness of 1. Big A, big B has a fitness of 2. And little a, big B has a fitness of 3. What's the best fitness you could have on this landscape? Yeah, this guy right, 3. And so if you were to start at big A, little b right here, we would expect to go from 1 to 2 to 3, but not necessarily, we would never go from 1 to 1/2, because that would be a decrease in fitness and an accumulation of deleterious mutations. So oftentimes in papers that are looking at more than 2 loci, you'll see crazy nucleotide sequence spaces that look like this. This is considered a rugged fitness landscape because it's very complicated. And on this particular fitness landscape, there are different features that I'm going to highlight. So first are global optima, which is the highest mountain in the entire place. So this is a particular genotype that maximizes your fitness. There are also local optima. So if, for example, I end up over here by random chance, that means that I can't escape from this local optima and get to the global optima without first decreasing my fitness, which I'm never going to want to do. So even though the best mutation possible is way over here, it's actually may not be possible for all individuals in the population to ever get there because you might end up on a local optima instead. And then fitness valleys are major decreases in fitness relative to the mean. And this overall point that I'm going to keep hammering home is that you can only go uphill, never downhill. You're always increasing fitness. Decreasing fitness is deleterious in the short term. And evolution is always acting in the short term. Evolution cannot know that in 10 generations, this mutation is going to be amazing because it can only see what's happening right now.
So in this case, I guess if evolution is acting in this space, there's really no reason to ever, and maybe that global optimum would never be reached because both ways you have to, sorry, you can go to the W25.
So
I didn't realize, I thought there were two. Is this a different thing?
Yeah, this is a different fitness landscape than the other one. This is a complicated one.
Okay.
But what I will say is where you end up on this fitness landscape is a function of where you started.
Right.
So if you start here, you can get either to the global optima or the local optima. But if you start here, you're never getting to the global optima because you're going to get stuck on that local optima.
Yeah, I was thinking about it.
You're thinking about it as the
previous one with the 1, 2, 3.
Yeah. Yeah. Sure. So deleterious mutations happen. Random chance happens. In a perfect world, you would never go down. There's some interesting papers that other people have done that looked at shifting fitness landscapes or how changing environments can lead to trajectories on these so-called rugged fitness landscapes. In general, though, if a mutation is deleterious enough in the short term, it's going to be very rare. And at least in this particular fitness landscape, it's not that you need to go take one step downhill. It's that you need to take one step downhill plus another step down, plus 10 more steps downhill. And that becomes increasingly less likely. So when we talk about interactions between 2 loci, it is possible that you can get there by random chance. When we're talking about interactions between 100 different mutations, it's really hard to get there by random chance. So the most uncomplicated version of this scenario is one in which mutations are additive when they're not interacting at all. So this fitness landscape would look something like this. This one is labeled correctly. So here, this is, AB is the wild type. And whether or not I have the little b locus or the big B locus, gaining the big A mutation increases my fitness by the same amount. It increases my fitness by a half. And similarly, whether or not I have the little a allele or the big A allele, when I gain the B locus, my fitness increases by the same amount, by +1. So in other words, the selection coefficient of both the A and B alleles do not depend on the genotype of the other one or the context in which they're in. And on this particular fitness landscape, if I started here and wanted to get to the global optima, both of these trajectories are possible, even though one is probably slightly more likely than the other. Epistasis is our word for when mutations interact in non-additive ways. So a more complicated version of this might look something like this, where now the fitness of one of our genotypes is not what we would predict. In this case, the B allele is beneficial in both cases, but it's way more beneficial. The relative increase in fitness is significantly greater if you have the little a allele compared to the big A allele. And if we were to start at this wild type little a big B or little a little b, we're probably going to end up at this fitness maxima regardless of where we started. So little b or the gaining the big B has a different effect depending on what the A allele is. There are 3 different types of epistasis that I was taught because my PhD advisor was this guy. But in the literature, you'll find others, and I'll explain kind of some of the other terms in a second. But so no epistasis or magnitude or additive genetic interactions. Again, you'll see this fitness landscape that looks something like this, where regardless of what the genetic background is or what the locus looks like at the other, or what the genotype is at the other locus, you get the same fitness effect. So you end up with, if you were looking at the fitness of gaining the, or of going from big A to little a, you have the exact same slope regardless of your genetic background, regardless of whether you're little b or big B. If I were to make the opposite graph, I would see the same thing. Magnitude epistasis refers to when the magnitude of effect changes, but the sign doesn't. So for example, a mutation that is a little bit beneficial becomes a lot beneficial, like the previous graph I showed you. Or a mutation that's a little bit deleterious becomes a lot deleterious. So this is an example of another magnitude fitness landscape. And if I were to make the same graph again, it would look something like this, where going from big A to little a causes the fitness to decrease in both cases, but the slope of those lines is changing. So again, magnitude of effect, but not sign is changing. And this is different from sign epistasis, where the sign is dependent. So in this case, you might see a fitness landscape that looks something like this, where going from little a to big A is deleterious sometimes, but beneficial other times. So in that case, you will see a fitness landscape that looks something like this, where for both the B, whether or not you plot it as little b, big B on the x-axis, or big A, little a on the x-axis, in this particular case, when you're going from getting the A locus, getting the A mutation, changes sign based on what your background is. But you'll notice for the B locus, it's not actually changing sign. The lines are crisscrossing, but this would, if you were only to plot one of these, it would look a lot like magnitude epistasis, which is why you kind of have to plot both of them. So again, if you think about going from little b to big B, it's beneficial here and really beneficial here. Reciprocal sign epistasis refers to when either way you plot this, the slopes are changing. So in this case, both variants are have a different sign when you encounter them alone versus when you encounter them together. So when you plot them, they'll look something like this, where A is deleterious alone, but beneficial together, and B is deleterious alone, but beneficial together as well. These shapes matter because they impact how accessible trajectories are. So if we imagine all of these different points are our starting points, if there's no epistasis, both pathways are equally likely. And so we'd expect to see a mix in a population of all 4 of these different genotypes, although most individuals would probably be at the optima. By the end of a simulation, we would expect everyone to be at this global optima. Magnitude epistasis, on the other hand, means that one pathway ends up being a lot more probable than the other. So AB are way more likely to transition. So you have one transition that is way more likely because in that first step, you're increasing the fitness a lot. Sign epistasis renders some trajectories completely inaccessible, while others are the only ones possible. And finally, reciprocal sign epistasis is the only one of these that is actually going to generate local fitness optimas where you'll get stuck and not be able to get to the global fitness optima in these very specific toy examples. So now in the literature, you might see examples, people defining epistasis like this. You can also see people defining epistasis in terms of positive or negative epistasis. In these cases, they're referring to the sign of effect when encountered together. So magnitude epistasis can be beneficial or deleterious. You can end up with a mutation that is very deleterious when alone, but a little bit deleterious when together, or the opposite of that, a little bit deleterious together, or a little bit deleterious alone, very deleterious together. And then the other options are a little bit beneficial or a lot beneficial together. When people are talking about positive epistasis, they are typically referring to alleles that are beneficial together, regardless of what their sign of impact is alone. Frequently, people who use positive versus negative are focusing for the most part on sign epistasis and reciprocal sign epistasis, because they're kind of the coolest and weirdest, and so they're the most fun to study. But if you're in the literature and you're like, what's positive epistasis? There's versus negative epistasis. It's kind of like another dimension onto this that refers to what is the selective effect after you encounter mutations together. Okay, so how would you model pairwise epistasis in SLIM? Inside of initialize, you would want to, I would personally want to define different constants, like what is the selection coefficient alone versus together. I also would want to define how far apart I want these two epistatic loci to be. And then I created other mutation types. But for M3, I'm saying that the selection coefficient is their S-ind, or the selection coefficient when alone. I also, because it's epistasis, and it is very important that I count genetic interactions. Okay, sounds good. With epistatic simulations, it is very important to turn convert to substitution off. In the SLIM cookbook, they have this example where they do epistasis, and what happens is one will reach high frequency and then get converted to substitution, and then the other one will rise to high frequency and get converted to substitution. So if I care about interactions, I obviously want to turn that off. I also created a mutation stacking group. This is a thing that I did weirdly yesterday. So here I'm creating a mutation stacking group that includes both M1 and M3 mutations. And then I'm saying that mutation stack policy is setting to last, so whatever is last. In the same way I did in the previous simulations, I'm creating different genomic element types, those in which there are no adaptive or no epistatic alleles versus a G2 region where I'm going to let those epistatic alleles be. The main difference in this recipe versus the last one is that now I'm allowing back mutations to occur. So now both neutral and beneficial mutations are allowed to occur at this locus. And then when I create the genomic elements, I create, again, very similar to the previous time, I'm saying G1 for some amount of length, creating a single epistatic locus. Then I am creating a gap between the first epistatic locus that is of a length of epidist. And then I am creating the second epistatic locus and then plugging in the rest of the genome. Obviously, this is also missing mutation rates and recombination rates when you do it yourself. Pop quiz, based on the S-ind and S-together values I have here, which of the three kinds of epistasis is this?
It's the third line. S-tog is S together.
You can call these whatever you like. I'm using symbolic constants here. This isn't like the P1 thing where you have to do it their way. Clearly, I am creating these symbolic constants, and so I'm calling them what I like. But Yeah, so they're both positive. So we know they don't differ in sign. So we know that that it's not sign epistasis or reciprocal sign epistasis. And if it was additive, what would we expect S together to be? So if they're both beneficial, they're not quite. We would just expect it to be S in times 2. So 0.002. Now, here I am still including burn-in. And then I'm allowing epistatic mutations. And then I ended the simulation after a certain number of generations. The epistasis part comes in with mutation effect M3. This is different than mutation M3, which is where we used the sim tags. Mutation effect, we're specifically modifying just the fitness. So first, I need to check how many mutations of type M3 you have. Because I created a mutation stack policy that is going to only count your last one, this is also ensuring that I'm not accidentally double counting a mutation at a single locus. You have to individually count both chromosomes. So right here, I'm saying, what is your count at genome 1 separately? And if it is greater than or equal to 2, it should just be equal to 2. I can specify a new fitness. So I'm saying if I get 2 mutations on genome 1 or on chromosome 1, we're going to return a new fitness value that is equal to 1 plus S together over 2. Here's where Slim is a little bit stupid. This return is in a fitness value, and it's going to give everything this fitness value and then add the selection coefficients later. So with this particular recipe I'm giving you, what you have to do is do 1 plus S together over 2. If you don't do S together over 2, you're going to end up with a really large, a selection coefficient twice as large as you think you will. And if you just do S together, or S tog, it's going to give it a very low fitness. So in the Slim manual, in their epistasis cookbook, they're doing like a lethal epistasis version where the mutations are beneficial alone but deleterious together. And so they don't care actually about any of this. But if you're trying to make them beneficial together, you start to run into issues like this. But then I also have to see if anything is happening on chromosome 2. So I would do something like the exact same thing, but switching out genome 1 to genome 2. And then else return effect, which it will just use the defaults. And that is the entire epistasis simulation. So going back to stealing slides from the class I taught last year on infectious diseases, a really interesting example of epistasis includes beta-lactamase resistance in E. coli. Beta-lactams are a really common type of antibiotics that includes like all of the penicillins, like penicillin, ampicillin are all beta-lactams. And E. coli can gain resistance to beta-lactamase through mutations at 3 loci. So if they like solve the structure, if any of you have gone to the AlphaFold workshop, this will look familiar. You can see that all 3 of these mutations are kind of near each other on the structure of the protein. So that's another way that we kind of think they might be interacting and making the structure more stable. And it's got a crazy fitness landscape that looks something like this. So to go through the annotation here, because it's kind of a lot. It's starting at the wild type and then here going to the triple mutants. Okay, those are the different genotypes. Each of these refer to an amino acid changing mutation. So this one is the P that is at position 21 is being converted to an L. If you've taken like biochemistry, these are all of the 1-letter codes. You're the chemist. Sorry, I put you on the spot. What does P stand for? What is it? No, it's not.
Is it like leucine or lysine? There's 20 amino, okay, so there's 20 amino acids, they all have one letter codes, and I cannot remember any of them.
P is proline.
Proline, thank you. So the proline that's at position 21 is being converted to a leucine,
Yeah,
or
leucine,
like
yeah.
leucine. Great. What's lysine?
Okay.
Okay. Sure. I mean, I know you're right. But this is why I really hated biochemistry. Okay. And then the A that's at position 26 is going to a T. L at position 28 is going to an R. These refer to the double mutants. They got both of them, but not the third one. And this is the triple mutant. And they did a really cool like experiment where they like modified the genome to make sure that they got all versions of this and then checked their relative abundances. And so these are the relative abundances given this key. So on this particular fitness landscape, what is the global optima?
Basing it off of the abundance.
Yeah, so use relative abundance, use that as a proxy for fitness. Yeah, so this is, so if you look at this, okay, so if you look at this particular graph, where this is step 1, this is step 2, and this is step 3. Step 1, step 2, step 3. So this is the starting point. These are the single mutants, these are the double mutants, and this is the triple mutant. Yeah, so that, which genotype is that on our graph? Yeah, exactly. So that's our global optima. So what do we think, what is the, so if we start at wild type, what is the first step you would take? Yeah. And then where would you go? That's true. So this is possible and this is possible. This one's slightly more likely because it's going to increase fitness more in the short term. And then you'll end up here, right?
Yeah.
But either whether or not you get the P21L or L28R, in both cases, the next step is the same. And so this is the most likely mutational pathway. Although really any of the first step ones are possible. Being neutral is okay. It's not great, but it's fine. Yeah, so if you accidentally ended up here, then you would probably at the next point go here, and then you'd be stuck there. Yeah. All right, so as our next practice, I would love it if everyone could try to code this. This is a hard one. So I recommend explicitly calling these each their own M mutation, M2, M3, M4. And use these as your fitness values. I've included like all of these genotypes at the bottom to help you figure out what it is. This is a really hard one. Why did I make you do this? You can use, you can use any positions or any size you want. I suggest you use theta equals 1, and if you do that, you do not need to make this go for very long, because these are huge selection coefficients. It should evolve really quickly. And you can make your chromosome kind of stupidly small. And I would estimate that these mutations are 15 base pairs away, and these are about 6 base pairs away. So
I'm gonna let you play with that for 10 minutes and then I'll show you the code I wrote. But at the end, my question is, did your so my goal for you is to code this all up, run it, and with these fitness values, it should be pretty clear. Is the most common genotype at the end of your simulations the global optima? You can use any mutation rate you want. I would suggest making it kind of high. Yeah, I would suggest making it pretty high. You can make just because you're trying to get a lot of mutations. You can make the mutation rate across the whole genome high. That's fine. The only downside of making the mutation rate across the whole genome high is that it slows down your simulations quite a bit.
You're trying to see data.
So you do not need to use the suggested theta. I'm just suggesting a high mutation rate.
The data would be not just for the.
Yeah, you can undo it either for the whole genome or for just one location. You can also make your population size anything you want, stop it at any point you want. For the most part, it shouldn't matter. I'm going to share the code I wrote now. I haven't updated my Slim to the newest version that kind of recently came out. So your things might be different. But here is where I defined all of my constants because I do love to use use those constants. And here I'm creating neutral and deleterious mutations for the background. I made P21L M3, A26T M4, and L28R M5, and I gave them their selection. So here, remember, this is their selection coefficients when alone. So that was the change in fitness from the wild type relative to the single mutant. Then I created all of the back, then I did all of the back mutation stacking, although that part's really not super necessary for the purposes of this. And then I created the genomic element types. So G1 region is kind of everywhere else where neutral and deleterious mutations are possible. Then I made G2 the P21 locus, G3 the A26 locus, and G4 the L28 locus. Then from 0 to 99, there's no epistasis. From 100 to 100, this is the P21 locus. No epistasis, A26 is at like 117. A lot of bookkeeping. And then I gave it my recombination rate and my mu, my very high mu. This is not load bearing. Mu is equal to theta over 4n, and I said that theta is equal to 1. Okay, I gave all 3 mutations their sim tags, although really fundamentally, if you didn't want to do a burn-in, that would be okay. I think for the purposes of this exercise, it's not as necessary. Now here's where it gets, gosh, this is so long. Okay, I created a for every single one of my epistatic mutations, I created a new mutation effect callback that is going to modulate their effect. And so for if individual genome 1 count of mutations of M4 is greater than 1, and nope, I'm just kidding. So there's multiple different permutations here. I want first is if the genome 1, if they have the M4 but don't have M5. Then if they have M4, then it's the same thing, but for the second genome. Then if they don't have M4 but do have M5, and finally if they have M4 up here. If they have both M4 and M5. And I did that for all of them. I'll upload this code because it's kind of a lot. And then if in the GUI, I press start. So you'll notice there are none of the beneficial mutations are occurring yet because I'm still in the burn-in. But right around now, the burn-in is starting, or the burn-in is ending, and some beneficial mutations are perhaps possible. Not yet. What is N? Now, okay, they're blue. So you can see this is the frequency of the different mutations. So you can see they're starting to occur. This is displaying haplotypes, which is, I think, the most useful thing to do. So if I stop this here and I create a haplotype plot, with, say, a sample of 100 individuals. This is super messy. There's a ton going on here because there are so many mutations. But I can actually turn off certain mutations. Oh, you can't see my, can you? Okay. So one thing I might do is say I actually only want to see the M3, M4, and M5 mutations. Now it looks something like this. So what is the most common genotype in the population? If this is a haplotype plot that is showing all of the 3 different mutations I care about right now, what it's saying is that the mutation that's at this locus is at a frequency of 100%. There's no one has the mutation that's at this locus, and everyone has the mutation that's at this locus. And if you look at the positions, this happens to correspond to what we would expect the global optima to be. I'll upload this to the course materials slide. Like I said, it's kind of a long piece of code to write because you need to independently write every single one. But you should get some, if you do in fact run it, you should get something like this, where eventually, especially when the mutational input is high enough, it converges on the fitness optima, and basically everyone in the population has it. So I'm gonna have, in the interest of time, I'm gonna take our break now and make everyone who's taking this for credit, which is everyone but Brendan, although if you want to take the quiz, Brendan, you can. Take the quiz. After you're finished doing the quiz, fill out the course eval, but also take time to take a break. The quiz is no notes, no phones. So close your computers and you shouldn't need a calculator. This is so the first eval. Do a quiz first and then do the first eval. Yeah. I only have one pen up here. I can ask you what more. There. Do you want to take a quiz? I don't know. I'm kind of seeing how you do it.
Okay, I'll take it.
You don't have to do it.
Yeah, so wait, I have to switch that off.
Yeah, pen or pencil, it doesn't matter. My goal is that it should not be that hard. That's what I have to do the quiz. And that they've been having problems with generative AI. And so they're pushing everyone to make their quizzes in person. I might be the first workshop that they're like, this is like It's just the 10 true false questions.
So what is what would you expect to happen? Yesterday.
So what would some of us use?
Well, yeah, what about some, the graph of, I chose a random one. I think it's Alice. Wait, yeah, this is like, you made this. Oh. So good on a lot of people. It's like, why do you remember what your graph looks like? And what the parameters were? I think it should be. I don't think. If you're done, you can just bring it up to me. I'm like hesitating to say that I remember the direction of this. Um, uh, in the answer key, I gave you the equation for FST. I'm gonna give you the equation for FST because I didn't need to do that. I'm not gonna tell you what it means, but look at that. I originally was going to make you calculate FST, but then I decided I didn't want to actually. Yeah, I thought it was easy to grade. Also, the class on infectious diseases I taught, my favorite question I ever gave was true, false, viruses are alive. Because that's a subject of debate. And I just accepted either answer. It was just to make people panic. Yeah, and so everyone had a little panic attack. It was like, I don't know what the answer. And like you could see them having little panic attacks on their answer key. They were like, they were like would write me notes. It was so funny. That's my favorite exam question. So. Ooh, Brendan. Here's what I'll also say with respect to grading. My goal is not to fail anyone who showed up and tried hard and learned something in my class. This is supposed to be kind of a metric of how much you're digesting. If you are worried about your grade, you can return in this to me with like writing on the back. I'll have an option to read to get a higher score if you don't like the score you got. I will say.
I don't ever see my grade.
I was very good.
I've gotten.
Yeah, I don't know. Great, thank you. Yeah, please fill out the course evals. Also use the restroom. And we'll wrap up the last hour. Brendan, do you have a red marker? Your grade is so, Brendan, you didn't show up to most of this workshop. You did sign up for the
It's like a whiteboard. I don't mind.
That's slightly better than what I had. Thank you. I'm gonna grade your stuff while you all write my course.
I have a red colored pencil better.
That would actually be slightly better. Thank you.
Grade your assignments.
I'm gonna go over the answers while Audie's gone.
Or I'm gonna go over the answers while Audie was gone. First of all, everyone here did great. You're all fine. Most everyone in the room, so only one person got everything right. So the biggest trick question was about the fitness landscape. That was true. That was an additive fitness landscape because it was actually the exact same one I used in my slides on additive fitness landscapes. 2 out of 5 people got that one right. But if you notice, if you go from this one to this one, it's 1 to 2 versus a half to 1.5. And then you go from 1 to a half or 2 to 1.5. It's both. In gaining either mutation gives you the same class effect. So that was the most missed question. That was the most missed question. I'll just go back to that slide really quick, actually. But yeah, this was the exact fitness landscape I showed you. And like I said, this transition increases your fitness by 1, and this transition increases your fitness by 1. So going from going from little b to big V has the same change regardless of background. And then the reverse is true. Going from here to here increases your fitness by half, and this increases your fitness by half. A lot of people, when they see a fitness landscape, they're immediately like, it's epistasis. And it is the most useful way of describing episodic interactions, but I'm not always going to show you that. Or you might not always see on a fitness landscape that the interactions are epistatic. Yeah. Like I said, please complete your official thing. Yeah. So the exit ticket link is on is on the slide, so you can do that afterwards. You're welcome. Awesome. Thank you. Brendan, you weren't here yesterday, and all the questions you got wrong were from yesterday.
Okay.
But when I was grading yours, you were the first person to get the fitness landscape one right. And so good job. All of the questions that were about things that you got correct.
Okay.
I mean, yeah, you're still in the lab. I'm not going to kick you out. Oh, I forgot to pause the recording. Eloy's going to be mad at me.
The other stuff,
I can send you my slides. It's fine. Or we'll just talk later. How many of you have run Slim from the command line before? Wow. Okay, good. I forgot to pause. Huh? So, I actually have never used STD PopSim, which is like a way of like importing ready to use parameters. I think if yeah. All right. I'm gonna keep going. I'm sorry to the recording and Aloy that I forgot to do this. I did go over the quiz. Well, okay. So you got the fitness landscape one wrong. It was an additive fitness landscape. Yeah, it was actually the exact one I used in my slides to just illustrate what an additive fitness landscape looks like.
No, I mean, the A, little a to big A and little b to big B.
So if you look at it, when you go from the fitness difference going from little a to big A increases by a half regardless of what your locus at B was.
Oh.
You can't pay attention to the heights. You have to actually look at the numbers.
I think I did just see your fitnesses to adjust.
Yeah. So you cannot see a fitness landscape and just assume that it's epistasis. That was my yeah. You just saw a fitness landscape and you assumed epistasis. That's how I get you. Okay. I'm going to talk about running Slim from the command line, but since basically everyone left in this room knows how to run Slim, I'm going to just do it really quickly. And I'll also talk about very briefly about some optimizing simulation stuff. But anyway, you can run Slim from the command line, and you should do that. But everyone in this room has done that before. So when you're running Slim from the command line, you do not actually have to define the values of constants. You can just call those constants in your script. And then when you run Slim from the command line, you just do slim -d to declare a constant value, and then mu equals whatever value I want it to be. So I'm never defining mu in this script, but I'm doing it from the command line. Bam, you're done. You have to rewrite D every single time. And then the quotes around, if you want to have one of your inputs be a string, are important and kind of funky, where you do quotes around the entire thing plus quotes around just your output. Don't do that, it freaks out. And then whatever your script is. Bam, that is how you run Slim from the command line. And it also allows you to make the same script be run several different times. Maybe like you want to run across a range of different thetas or a range of different S-bends. Yeah, pay attention to quotes around file path. This is a minimal bash script to run on Hoffman. Slim is already on Hoffman. Is Slim 5 on Hoffman? Okay.
No, actually, you have some code that
Okay. So Slim 4 is definitely on Hoffman. If you're trying to use Slim 5, there's some weird stuff and you should talk to Addie about it. Yeah. I defined an output directory. I like to make the job ID. Yeah. And then here I'm running 10 different replicates. And I'm making an output file called outputdir.slimoutrep_i. And each replicate, I'm randomly drawing a new selection coefficient. It's beneficial every time, but it could be anywhere from 0.01 to 0.05. And then this is the same thing I just showed you. So this is a very minimal bash script to run on Hoffman 2. You can also do things that are a lot more advanced. Then optimizing simulations. This is the actual code to do for loops in Slim. I showed you the wrong thing yesterday. But one of the reasons why Slim uses this like P1, M1 like syntax is so that you can create things inside of for loops. So let's say you wanted 500 different subpopulations. I know Audie is doing those kinds of simulations where you've got like a million subpops. You do for I in 0 to however many I want, sim add subpop I 500. So it'll automatically convert add subpop 1 to P1. And then if I want to set migration, this particular recipe from Slim is then setting migration rates between all of these and saying that migration is occurring basically unidirectionally. So with setting subpop I, so subpop 1, set.migration rates, the subpopulation before it immigrates into that subpopulation at a rate of 0.2. So this is kind of like a one-directional stepping stone model. One way of optimizing simulations is to reuse your burn-ins. I know some people who will like write one burn-in and then reuse that for 20 different replicates later on. Or if you're doing a case where you're trying to do an add neutron mutation and want to see if it reaches fixation, if it doesn't, you can just return to start. So this is a very simple recipe where at the end of your output, where at the end of your burn-in, you just output the full, you say sim.output full. In this particular recipe, I'm taking a target, sampling that target and adding, and then giving that target a brand new drawn mutation, which is what Yashis was talking about earlier. Then this is, I mentioned this before, but in this particular recipe, I didn't use the convert to substitution trick. So now instead of something reaching fixation, it's going to reach a frequency of exactly 0. So if the frequency of M2 is ever 0, I'll first check to see if it actually is because it's substitutioned, in which case it fixed, and then I'll say the simulation is done. But if not, if it went extinct, I can read from the population file and it'll just go back to that point. An important point here is that if you read from the population file, it is going to also set the generation to, or the tick, I should say, in Slim lingo, to whatever it was when you left off. Then I set a new seed, target a new sample, and then it'll essentially just start over. And so this is one way of reusing burn-ins and making sure that every single replicate ends in fixation. Now you're only saving replicates that end in fixation. How many of you have heard of population size scaling before? Okay, so in our class, we've talked a lot about population size. What happened when you use a small population size in your simulations? What happens? Huh? Drift. Yeah. What else? What other things happened? Speed. They're faster. You're keeping track of less individuals. It's doing fewer computations. So a really common practice in simulations, not only in Slim, but it's become really common in Slim, is to use what's called population size scaling. So many of the parameters that we've already talked about, like theta or even selection coefficients, are communicated in terms of n. And also the burn-in is in terms of n. So if we put parameters in terms of n, we can scale them up or down. So let's say we have a starting n of 10 to the 5th, but we want our simulation to run a little bit faster. We have mu of 10 to the -7, recombination of 10 to the -7. S del is -0.001, S ben is 0.01. If we wanted to rescale all of this to be a smaller population size, say 10 to the 3rd, we would use a scaling coefficient of 100. So now, in other words, I'm saying n0 equals n new times q. And that means that I can do this little trick where I multiply everything by q. And that should give me a population scaled version of parameter space. So now population size is equal to 10 to the 3rd. Mutations are occurring more often. And then selection coefficients are bigger, right? Because as population size gets smaller, you need larger and larger selection coefficients in order to need to be seen. Here's a paper from earlier this year where they looked at the effect of scaling in Slim specifically, and they used scaling factors of 1, 5, and 10. So 1 is obviously no scaling, at 5 and 10.
on different simulated chromosome lengths. And this is showing simulation times in minutes. So as you can see, as you make your population smaller, as you increase the scaling factor, the simulations run a lot faster. But Emma's already highlighting kind of my note of caution here, which is that population size is not a non-parameter. Smaller population sizes are impacted by drift. Ferrari also goes into this, but I think the figures in Dobby were a little bit nicer to put in slides. A couple of new papers that, like, again, have only come out in the past year or so have found that population size scaling can introduce errors into your results for the reasons you think, where small population sizes mean random chance is going to have a larger impact and drift is going to be more important. So this is mean percent error of allele frequencies and mean percent error for fraction of mutations fixed as a function of scaling coefficients. So mean percent error is a measurement of how different the distribution is from what it was when there was no scaling used. And you can see that for the fraction of mutations fixed, as we increase the scaling factor or decrease the population size, far fewer beneficial mutations are reaching fixation compared to what we expect. For equilibrium allele frequencies, it's kind of all over the place, but you can also see that as you increase the scaling factor, as you decrease the population size that you're using, errors are increasing overall. So the smaller your scaling coefficient is, if you're using one, the better. A lot of people use a scaling coefficient of like 10, which can introduce a lot of errors in your results. And then I guess one of my last slides is that SLIM has a lot of built-in output utilities that you can use. Output full is what we've mostly used so far, which outputs kind of that default SLIM output for all individuals in the population. But you can also do output sample, which takes does that output, but only for 100 individuals or whatever you say. You can also output into a VCF. So if you are, when I was doing KNID PopGen, I was working with a ton of VCF files. And so if the data that you're trying to match looks like VCFs, that might be useful. Output MS sample is also a really useful tool. That's MS sample is going to give you essentially like a CSV, or it's not a CSV, it's a .ms file. But it's a matrix that has all of the genotypes where zeros are ancestral and 1 is the mutations, but it doesn't have any information about sin or non-sin or mutation type. So it's not perfect and is missing some information. But if you don't need to know about what mutation types are, it's useful for just understanding patterns of diversity. And for both VCF sample and MS, if you don't include the sample part, it'll just output the full population. You can also use specific outputs like output mutations, which just does the mutation part of the default output. Fixed mutations, which is only mutations that have been fixed or converted to substitutions. And substitutions. Anyway, so hopefully today you have learned some basic, or not today, over the course of the last 3 days, you've learned some basic PopGen theory that can help you develop intuition around your expected results, which is a really important part of debugging your simulations, is knowing what you expect to see and knowing how to get outputs that will tell you whether or not this aligns. We talked about demography and the importance of population size, as well as types of selective sweeps. And you've also learned how SLIM leverages Wright-Fisher reproduction to model genetically explicit genomes in a forward in time simulator. And yeah, so here's your last exit ticket. Again, this is different from this is a second Google form for you to complete. But this is your last bit of homework assignments. And I'm going to end the recording here because they don't need to